Hello everyone, welcome back to another Watershed walk. It's been a while. We've just wrapped up our Ripples and Roots nature camp last week, which was a blast, but now we can get back to some of our regular programming. In this video, we revisit the installation of EAB and HWA traps back in May. These traps are meant to monitor for invasive species, specifically emerald ash borer, that's EAB, and hemlock woolly adelgid, HWA, both of which can decimate native tree populations.
Top of the trap, and I just take this here and I put this in the middle so it hangs from the middle of the trap so that when they try and fly towards it they get stuck on the outside of the trap. And these go in here like that. And then this is where the branch is, and it hangs from the tree like that.
I had the privilege of tagging along with Laura and our summer students to search for a spot for each of the traps. Basically, a hemlock tree and an ash tree, preferably within a stand. The hemlock tree was easy to find. The ash tree was surprisingly scarce. We climbed uphill, over a ridge, back down, up another hill, and back out to the road before we spotted one. We have been using the EAB monitoring traps in partnership with NBISC for a few years now. So far, we've not found any evidence of emerald ash borer, but we continue to monitor for it so we can take immediate action when necessary. The HWA traps are new to us this year and are not so much traps as DNA collector plates. Placed beneath the boughs of a hemlock tree, the plate collects DNA from pollen particles. If the lab analysis finds hemlock woolly adelgid DNA, we will take next steps to mitigate the threat to our hemlocks.
We're here at our second hemlock woolly adelgid trap. We are along the bluff in a hemlock stand. So this is an ideal spot since there is a large hemlock population. And we're just going to take the slides, which are covered once again in Vaseline. We're going to take them out using gloves so we don't infect them with our own DNA. And then we will place them into a tube that will then be analyzed for DNA. So the goal is that there is no hemlock woolly adelgid present since it is an invasive species, and it will lead to the downfall of hemlock, sadly. Sadly, there's a lot of forest pests as climate change progresses and as the world becomes more connected and people transport things from place to place, making it really easy for an invasive species to spread. So we're just going to place the slides back to back with the petroleum jelly out. And then we're just going to slide it into our test tube. And then we're going to do that with the other one, so there's 4 little slides on each trap.
Thanks to our partners, New Brunswick Invasive Species Council and Canadian Rivers Institute for your continuous support and equipment, and to our funders for making the work we do at the HWA possible. Don't forget to like and subscribe if you're interested in seeing more content like this, and follow us on social media to keep up with our environmental endeavors. Catch you next time!